And so if you think of scales, the quote unquote smallest scale, and I don't want to call it smallest meaning least meaningful, it's smallest in terms of size, is the genetic scale. And that's when people think of genes and how basically people may be pre-programmed to have certain types of biological processes, et cetera. So that leads us to our first set of panelists and first set of quick mini talks. And the first is Denise Ney. She's from the University of Wisconsin-Madison. She's a professor in the Department of Nutritional Sciences at the University of Wisconsin-Madison, where she served as department chair and director of the interdepartmental graduate program in nutritional sciences. She has conducted research on how nutrition impacts gastrointestinal physiology and rare genetic diseases for over 25 years, focusing on the neuroendocrine regulation and intestinal adaptation of PKU. It's an inherited metabolic disorder that requires lifelong dietary restriction of the amino acid phenylalanine to prevent cognitive impairment. She's recognized as a quote unquote rare disease hero for her research to develop GMP medical foods for PKU by the FDA Office of Orphan Products Development, as the recipient of the Spitze Land Grant Faculty Award from the College of Agriculture and Life Sciences, and Mary Swartz Rose and Fellow Awards from the American Society of Nutrition. She's mentored 13 PhD students with her research program, many of whom hold faculty positions, and directed the didactics program in dietetics, leading to over 1,000 students achieving careers as registered dietitian nutritionists. And she's published 135 articles, 10 book chapters, and inventor on 2 patents. She's received her PhD in nutrition science at the University of California, Davis. So obviously a luminary in the field. I'm going to introduce the other 2 panelists first as well, and then we can proceed to the talks. Jim Caput from Vidient. He's co-founder and chief scientific officer of Vidient, which is developing a comprehensive knowledge base of factors affecting health and disease with digital health tools to deliver personalized recommendations to individuals. He's been developing strategies and methods to target nutrition for improving personal and public health for his entire career. He previously was a biochemistry faculty member at the University of Illinois College of Medicine, director of Northwestern University's biotech laboratory, and he's held a number of other different positions. Each of the speakers' entire bios are listed on the website, so I'm paraphrasing some of the bios because they've all have had very impressive backgrounds. He's received his PhD from Colorado State in biochemistry and molecular biology, and spent 5 years as postdoc fellow and assistant professor at Rockefeller University. And our third panelist for this first session is Ahmed El-Sohaimi, and he's a professor and Canada Research Chair in Nutrigenomics at the University of Toronto. He joined the faculty at the University of Toronto in 2000 to establish a research program there in nutrigenomics. The goal of his research is to identify biomarkers of dietary exposures and elucidate the genetic basis for variability in nutrient response and dietary preferences. He's had a number of different talks, and he's editorial board on he's on the editorial board of 8 journals. He received his PhD in nutrition sciences from University of Toronto and completed a postdoc fellowship at the Harvard School of Public Health. So without further ado, I'd like to pass along the virtual mic to Dr. Ney so she can start off our session.
Thank you for the kind introduction, Bruce, and I am pleased to kick off the genetic scale panel. My topic is population newborn screening and the use of sequencing. I'm having some trouble advancing the slide here.
Denise, click on the slide.
Okay. Okay, thank you very much. Okay. By way of introduction, I'd like to talk a little bit about phenylketonuria, or PKU. This is an inborn error of metabolism caused by over 800 mutations in the phenylalanine hydroxylase gene. And PKU is truly the poster child for precision. I'm not talking personalized nutrition, but I'm talking about precision nutrition, where we have a strong evidence base. that a certain diet matched with a certain genotype is essential. In fact, the phenotype of profound cognitive impairment due to untreated PKU is thankfully rare in the developed world. The tragedy, as we see, of the untreated brother and his treated sister no longer occurs very often in the developed world. This is because of mandated newborn screening programs and initiation of the low phenylalanine diet. This was a diet that was devised in 1953, well before the genetic basis of PKU was understood. My topic for today is a recent study which discusses the role of whole exome sequencing, or WES, in newborn screening for inborn errors of metabolism. WES consists of sequencing the protein coding regions of genes. This is thought to constitute only 1% of the genome, but this is where most known mutations that cause disease are thought to occur. Newborn screening for inborn errors of metabolism provides an ideal model for evaluating the role of sequencing in population screening. The sensitivity and specificity of WES can be compared directly with those of tandem mass spectrometry, or multiplex MS-MS, which is the established technique for newborn screening programs. This study analyzed variants within an exome slice of 78 genes linked with 48 inborn errors of metabolism ascertained by newborn screening in California using archived dried blood spots from 4.5 million births between 2005 and 2013. For population-level newborn screening, it is imperative to identify all positive cases of inborn errors of metabolism, because many of these are life-threatening, while minimizing false positives to reduce undue stress on the family as well as medical resources. The sensitivity of MS-MS screening was 99.0% with a specificity of 99.8% in this California data set. And this is consistent with the quality control maintained by the CDC for newborn screening programs in the United States. The first conclusion is that WES alone is insufficiently sensitive or specific to be the primary screen for most inborn errors of metabolism. The sensitivity of WES was 88% overall. Remember, that compares with 99.0% with traditional newborn screening. 571 cases were identified with WES, but 103 cases were missed. The sensitivity, however, varied among the different inborn errors of metabolism. It detected 100% of methylmalonic acidemia cases that were caused by known genetic mutations, and 86% of the fatty acid oxidation disorder, VL-CAT. The specificity of WES was 98.4% overall. 11 cases were false positives. This would translate or extrapolate to 8,000 cases per year based on 500,000 infant births per year in California. In contrast, MS-MS yielded 1,362 false positives in 2015. Thus, WES is not as good in newborn screening as the first line. The second and last conclusion is that WES has advantages as a secondary test for infants with abnormal MS-MS screens before undertaking further biochemical and clinical studies. WES reduces false positive cases as a second-tier test. For example, of 108 false positive VL-CAD cases, WES identified 48 or 44% as true negatives. And of 16 maple syrup urine disease cases, WES identified 100% or all 16 as true negatives. Thus, WES would have avoided the cost and family stress of follow-up testing for, in this case, VLCAD and maple syrup urine disease, if the WES was provided in a timely and cost-effective manner. WES also has the advantages that it may reveal new genetic variants and suggest the need for whole genome sequencing. In fact, diagnostic whole genome sequencing is being used with success for critically ill patients with unknown diagnoses in the ICU. And in our discussion, perhaps we can talk about the use of whole genome sequencing and its application. Thank you for your attention.
Sorry. I'm Jim Kaput. I'm the co-founder and chief scientific officer of a for-profit company, Vidient. Even though we're talking about genetic factors here, I want to make the point that genetic factors are one component of the overall set of factors that influence health. And in fact, this information is widely available, although siloed in 32 million PubMed abstracts and clinical reports and gray literature. And what our company is doing is has created a transformer-based language representation pipeline to extract directional relationships between factors and individual health. While those of us who are born in the biochemistry lab always like numbers and believe in that type of science, there's huge amounts of literature that are available in the literature that you can extract information and use either to design better experiments, or what we're going to be doing is to deliver equitable platforms to provide individuals of all socioeconomic classes the information that's in that literature. But for this talk, we're going to talk primarily about the genetic component. We had a project that was completely funded by Nestle, that was done in Brazil, and we published a series of papers on that project. The very first one showed that a person's genetic ancestry was responsible for at least a portion of the levels of thymidine monophosphate for Europeans, for vitamin B12 for Native Americans, for folate in Native Americans, and also for the response to the intervention in Native Americans. This follows what Constance Hilliard said yesterday about the need to look at the genetic background of the individuals in order to understand how a nutrient, in this case we had a multi-nutrient intervention, the individual would respond based on genetic ancestry. While this was important, it wasn't as fine enough detail as we need to really do personalized information. So we've turned to the use of polygenic risk scores, because during my career in this, I've read huge numbers of papers in the genetic field and as well as in the nutrition field that indicate that a single SNP does not have an effect size that actually can explain a response to a nutrient or a susceptibility to a disease. So what's happening in both the genetic community and the nutrition community is they're using polygenic risk scores, which are combinations of SNPs that are available in the public domain in order to understand either a response to a nutrient or a disease susceptibility. There is no formal guidelines for how you do this. Each group does it their own way. They usually rely on GWAS identified SNPs or candidate SNPs in a pathway. And we know from what Dr. Constance said yesterday is there is a problem with the GWAS SNPs because they are largely Eurocentric. What we've done is to use this is a very complicated slide, but ignore the numbers. And what we've done here is we've taken a participant DNA and we've genotyped it with 5 million SNP arrays. And then we also did whole exome sequencing at a level of 70 to 100x. We then determined the genetic ancestry of each individual using the ancestry informative markers from the Human Genome Diversity Project. So that combined the genetic information from individuals and population. We then looked what we call a middle out approach, in which we took the SNPs that are in 90 genes that are involved in the genetic pathway of vitamin B12 metabolism. And we analyzed where those B12 genes were in the chromosomes and what each individual, the total in population of our children, 9 to 13-year-olds, what percentage had a higher percentage of European alleles or African alleles or Amerindian alleles. And what we found is that we could identify through data-driven approaches 36 SNPs out of this B12 that could be used for the polygenic risk score analysis. Each one of these bars is one individual, and the polygenic risk scores goes from low to high, and the colors of this green, light green, darker green, and blue represent the level of vitamin B12 in each kid. So you can see that the polygenic risk scores that are high have a preponderance of individuals with high B12 levels at baseline. This has recently been published. So this polygenic risk score explains about 36%, about 42% of the phenotype, which is, I thought, very high, but given that there are a lot of other factors involved. But the reason that we could get that number is because we had the covariance for each individual. We did sex, BMI, age, their total healthy eating index. So we believe in dietary patterns, and also the mean genetic ancestry of each child at each SNP that we studied. That is, we corrected for the genetic background at each loci that we use for polygenic risk scores. This eliminates one of the problems of population stratification, but it also eliminates the problem that we were heavily reliant on the genome-wide data that's in the Genome-Wide Association catalog at NCBI. So as just a proof of concept or pilot, how would you use this to recommend diets? So that we stratify the individuals into terciles based on their polygenic risk scores of these 36 genes. And we know that if they have a polygenic risk score that's high, their B12 levels in plasma at baseline were high, and therefore they're at low risk for needing additional vitamin B12. If they're intermediate, we have to, there's no need for additional B12, but we might want to improve their overall diet because their HEI scores are low in this study. And then in those that are highest risk have the lowest polygenic risk score, and they need to be followed more carefully, and you can make specific recommendations for their dietary intake. So this is one vignette for how you would use polygenic risk score analysis in order to identify what an individual would need for their dietary intake. So the take-home message is that we have to assess multiple variants rather than a single SNP. A single SNP in one gene and one pathway cannot give you the overall information necessary to recommend a diet. You need to understand the genetic architecture. In this case, we corrected for Amerindian, European, and African ancestry, which are the predominant genetic profiles in the Brazilian children. I want to just note as in passing, we got a lot of criticism for using an admixed population, but in reality, we could use that admixture to our advantage in a relatively small sample size in order to get solid scientific data. And then the analysis of the associations was corrected by things like the healthy eating index. So we took into account how much the individuals are eating, age, sex, and we could put socioeconomic status and other factors that are known to influence health. In this case, we focused on the pathway variants, this middle out approach, and therefore the 42% was quite good. We obviously missed some heritability, but I believe the rest of the heritability are things that we did not measure, like their exact composition of food intake, although we did food frequency questionnaires and 24-hour intakes for every child in the population. And of course, our results need to be replicated, but that should include covariants used in this case. So some of the SNPs that we identify because of the genetic population may not appear in other studies, but we believe that by doing the same similar type of study, we can then collect all the SNPs and use them on a genetic population specific basis. So that is what I wanted to discuss, and looking forward to a discussion on all of these points.
Thanks, Jim. So we only have a few minutes, and so apologies for having to move things along, but we wanted to squeeze in a little bit of a discussion at the end.
All right, thanks very much, Bruce, and hello, everyone. I'm Ahmed El-Sohemi from the University of Toronto. I'm also the CSO of Nutrigenomics. And so with that, as a disclosure, I am the founder and I also hold shares in the company, which provides genetic testing for personalized nutrition to healthcare professionals. So our main interest really is in the role of genetics in nutrition, is recognizing that if you trace the path from virtually any nutritional factor to any health outcome, if there have been enough observational studies, we know we see this heterogeneity of response. And there have been a number of pretty good examples now of specific genotypes that can help us predict if the intervention is going to improve our health outcome, have no effect, or hinder it. So clearly, one size does not fit all, and I think we can all appreciate and recognize that. Some of the earliest references to this, of course, comes from the 1st century BC, where Lucretius noted that what's good for one person might actually be harmful for someone else. And when we consider where or how genetics can influence our response to diet, genes influence our sensory perceptions and our appetite, some of the dimensions that were discussed in the session yesterday. When nutrients and food bioactives are ingested, genes or the gene product, whether it's an enzyme, receptor, transporter, impacts the metabolism, absorption, cellular uptake, digestion, and elimination of virtually everything we ingest. So our interest from a research perspective is really on these genes that impact response to nutrients and not specifically the disease-associated genes. And I think that's a very important distinction that we need to make, and I'll touch on that a little bit later on. So I just want to give one quick example of how a single SNP can actually be quite impactful. Some of what I'm going to say right now might sound like it's contradicting what Jim just presented. Jim and I have had many friendly discussions about this over the past decade or so. And this is an example from a 2-year randomized controlled clinical trial from Lou Q's group at Harvard, where they showed that a high protein diet impacts body fat and body composition only in those with the AA genotype of the FTO gene. Now, this is a single SNP. Not a SNP that predicts obesity or BMI, but predicts response to a high protein diet. This has since been replicated in a number of other studies, including a study that we conducted in a multi-ethnic population here in Toronto. And all of these studies show the same thing, that a high protein diet affects body composition only in those with the AA genotype, a single nucleotide polymorphism. Now, some of the comments, I think, and the controversies in this field, and I just want to go through some of those items. And I'll start with the first one, that Jim mentioned, that single SNPs are not that effective. And again, I think a lot of these comments and criticisms come from genetic association studies, looking at specific phenotypes like blood levels of vitamin B12 or blood levels of 25-hydroxy-D. Those markers, what they don't tell you is response to the intervention. So you can identify either a single SNP or multiple SNPs or create a genetic risk score that predicts existing blood levels of a micronutrient. What it doesn't tell you is whether or not those individuals are actually responsive to it. It's quite possible that those who have low levels might actually be resistant to the response of dietary intake. I've just given one example, FTO and protein, but there are other examples, CYP1A2 and caffeine, vitamin C and GST, and a number of other gene-diet interactions that are quite robust using a single genetic variant. Some argue that people won't change their behaviors, and I know John Mathers touched on this yesterday. There have been now a number of RCTs showing that giving people the right kind of genetic information coupled with actionable recommendations does in fact motivate people to change their behaviors, and that's been sustained for over a year. Some argue that we need more evidence and we need them from RCTs. Again, that evidence is now emerging. Some argue that results of genetic tests are too complex and people won't understand them. Well, if you give someone a raw readout of their genetic information from a sequencer, obviously they're not going to understand that. But to tell someone you have the genotype that makes you respond to a high protein diet, if you're seeking to lose weight, you'll benefit from upping your protein intake. That's not very complex. Some argue that family history is more informative, but we know that families share not only a common genetic background, but also common environment. And now there are a number of examples of how family history is just too crude of a predictor, and you really need to have that specific genetic information because you don't know how each of the offspring have actually inherited which combination of specific genotypes that might enable them to respond to a particular dietary intervention. And some argue just, follow recommendations for healthy eating. So if we do that for a number of examples, we'll see that that information is actually harmful. So just to wrap up, to address these controversies that, I keep hearing over and over again, and they often come up as questions when I present some of this work at various conferences. We published this paper just about 2 months ago addressing not only the advances that have been made, but also some of these controversies that I've identified in some of these boxes here, and to show that really genetics has advanced quite considerably. And while we see a greater rate of growth, as Mariette mentioned, in some of the other technologies like the microbiome, it doesn't mean that genetics has become obsolete by any means. In fact, the scientific evidence base is much more robust for genetics than any of the other omics technologies. So I'll just leave you with this and just kind of point out some of the points that have been discussed and that will continue to be discussed over the course of this workshop. I just want to emphasize that the evidence is robust, but for some markers. So it's important to know where you can give recommendations and where you can't. Haven't really had time to get into the differences between ethnic groups, but those for some of the markers, again, have been replicated. Greater compliance, information is actionable, and there's also evidence that this kind of information is more effective if it's given through healthcare professionals rather than direct to consumer. So I'll stop here. Thank you, and hope we have time for some questions.
All right, thanks, Ahmed, and thanks, Jim, and thanks, Denise. It's always a good sign when there's more information than time allotted. So apologies for having to move things along because we wanted to stick to our 11:15 start for the next panel. So that gives us just enough time for one question. So why don't I ask all of you a question that came from the audience. So the question is, how far away are we from evidence reaching a point for payers to be able to reimburse such solutions and hopefully level equity? Where is evidence strongest and weakest on efficacy currently? Any thoughts on that question?
Yes, I would say in the area of metabolic diseases with known or suspected genetic basis, the insurers will start to pay. In PKU, where I work, when I first entered the field about 10 years ago, I was surprised that most people didn't know their genotype who had PKU. And the answer why is because their insurance would not pay for it. But now it does pay to some degree, depending on your insurance.
Thanks, Denise. Jim and Ahmed, any thoughts, additional thoughts?
Yeah, my view is that they're not going to pay until we get more evidence. And I don't think the evidence is going to come just from a microbiome test or just from a genetic test. I think it's the integration of the data across multiple scales that will provide the type of information. And that means we're going to have to use machine learning tools in order to extract that information to make it solid enough to convince insurers and payers that the data is real and reliable.
I think whether it's a genetic test, a microbiome test, a platform that integrates various omics technologies, I think payers want to see a return on their investment. So the kinds of studies that need to be conducted are the economic analyses. And we've seen these in the field of pharmacogenetics, where there have been savings in terms of the additional costs associated with adverse drug reactions and reducing that. And that's where the payer will recognize that paying for these kinds of tests will actually save them money in the long run. And that's the kind of work that I think needs to be done.
Great, excellent. Well, wish we had more time for more questions, but the clock beckons. So again, I want to thank each of you for your presentation. You really gave us some good food for thought, so to speak, for the rest of the day. And certainly it's a good on-ramp to the day 3, where we're going to be talking about broader issues like regulatory issues and dissemination, et cetera. So this question actually ties in with some of the things that we'll be discussing tomorrow as well.